Okay, thank you very much. My statement, I have no financial or commercial interest to disclose. And I'm going to take just a moment to tell you a little bit about San Diego Zoo Institute for Conservation Research. It was founded in 1975 by an MD pathologist, Dr. Kurt Benerschke, who many of you probably know, who recognized a unique opportunity for comparative pathology research that was afforded by a large zoo. Since its inception, the Institute, with a small staff, the Institute has grown to include 8 research divisions with nearly 250 scientists and staff working in over 100 plant and animal conservation projects in about 35 countries. But today, I'm going to concentrate on just one specific reproduction project. So we have a very long history of breeding rhinos the old-fashioned way. And you can see we've been very successful, especially with the Southern White Rhino, with 100 births and 2 more on the way. Greater One-Horn Rhino with 71 births and 2 more on the way, and the Eastern Black Rhino. But we have never been able to reproduce the Northern White Rhino. Despite having 8 Northern White Rhinos over the years in our collection at San Diego Zoo Safari Park, the very few matings that were achieved after hormonal stimulation of the females were unproductive. The reasons for their infertility were unknown at the time, and although we have figured out most of those and taken subsequent action and resolved them, it certainly was too late for the Northern White Rhino. This species probably was never very numerous. We don't have good records prior to the turn of the last century, but you can see as these species declined in the early 1900s, 2,000 to 3,000 in 4 North African countries. And then by 1984, only 31 left in 1 country. In 2007, the last male was killed. They became extinct in the wild. There were 10 left in zoos, in 2 different zoos, one San Diego and one in the Czech Republic, the Králové. Unfortunately, only one of those captive females ever reproduced. She gave birth to 3 Northern White Rhinos and 1 Southern White, Northern White hybrid. One of her daughters is Najin, pictured here, who is now 31 years old. Najin gave birth to a single offspring, her daughter Fatu. They are the last 2 Northern White Rhinos on the planet. In 2009, 2.2 in animal speak and zoo speak is 2 males, 2 females, were moved from Europe to Opegeta Conservancy in Kenya. Those 2 males, Suni died in 2014, Sudan in 2018. But following the death of Suni and our last male, Angelifu, in 2004, Sudan stood as the last male Northern White Rhino on Earth. But the publicity surrounding these animals and the death of these animals really brought extinction from an abstract concept to a real-time certainty. We're literally watching this species go extinct. And for a very large, charismatic species like the rhino, this has had a tremendous impact on the public. So extending our long-term commitment to rhino conservation, San Diego Zoo lent its support to really an open-ended project that we named the Northern White Rhino Initiative. And in 2015, we co-convened a meeting in Vienna, which was appropriate. It's the oldest zoo in the world. The meeting was called Conservation by Cellular Technologies. And we published a paper in Zoo Biology called Rewinding the Process of Mammalian Extinction. And in addition to establishing these international partnerships at this meeting, the meeting resulted in this proposed roadmap for rescuing the Northern White Rhino. If we can successfully implement this, it would ultimately lead to our goal, which is a self-sustaining population of Northern White Rhinos back in native habitat. So I'll go over this very briefly. It looks fairly complicated. On the left-hand side in blue is what we call the constructed gamete side of the equation. Those are gametes that we would produce in vitro from originally fibroblast cells from our frozen zoo, transformed into iPS cells, and then primordial germ cell-like cells, and then with great effort and a lot of hope, oocytes and sperm that we could then put together in the lab. On the other side, the natural gamete side, is where I am primarily working. So to collect actual natural gamete sperm and oocytes, and either postmortem or from living animals. And I was very glad to hear that Pierre talk about postmortem collection. This is a very important method of collecting samples, and probably 75% of the samples that we have in the freezer, especially sperm, are collected postmortem. But we can go through, we're developing the techniques to create the embryos in the lab, to do in vitro maturation, in vitro fertilization through ICSI and embryo culture, and then embryo transfer. So if and when we can create sperm and eggs from fibroblast cells, they will come into our lab and we'll go through this process to create northern white rhino embryos for transfer into southern white rhino recipients. And if you notice that I said that there was one hybrid calf produced, northern white rhino, southern white rhino, that gives us hope that the southern white rhino is a very good recipient for northern white rhino embryos. So the first question we had to ask is, how related are these animals? Are they, we did have a hint that they could hybridize. Were they species? Early work with mitochondrial DNA analysis indicated that they were separate species. Some people say they're really not separate at all. They're just different geographically. They're just races of white rhinos, or they could be subspecies. So we pulled our fibroblast cells from our frozen zoo, and very, as Pierre mentioned, very similar to their frozen zoo. We have fibroblast cell lines, sperm, eggs, embryos, testicular tissue, ovarian tissue, stem cells, pure DNA, and we have added seeds and plant cultures to the frozen zoo recently. So we pulled those northern white rhino fibroblast cell lines. We have 12 northern white rhino cell lines. And you can see here in A, the geographic distribution of northern white and southern white. Northern white, of course, in red in the north of Africa, and southern white in South Africa. In B, we see the admixture results. This is 9 northern white rhinos and 4 southern white rhinos. And you can see that there's very little admixture here, in addition to the one hybrid, known hybrid in current times. By principal component analysis in C, here you can see that the northern white rhino is clustered very differently than the southern white rhino and has more diversity. So sort of simplifying this, here are some estimates of genome-wide diversity in northern and southern white rhinos compared to other mammalian genomes. So we see that if you look sort of in the center, human heterogosity is less than southern white rhino and quite a bit less than northern white rhino. So the final knowledge from all of this genetic analysis is that the northern white rhino in the 12 cell lines that we have in the frozen zoo is we have greater genetic diversity in those cell lines than we do have in the current southern white rhino population. The reason being that the southern white population went through a couple of very severe bottlenecks. The white rhino did not until recently, on its way towards extinction in the wild. So what about these fibroblast cell lines? So when they're first thawed, they're first collected, they're karyotyped, and 9 of the 12 cell cultures now have been turned into induced pluripotent stem cells. And the question, of course, is can they differentiate? And the answer is yes. When they are allowed to differentiate spontaneously, they do produce all 3 germ layers. And they might also spontaneously, probably with some additional in vitro genetic manipulation, but they should also spontaneously generate primordial germ cell-like cells on their way to sperm and eggs. If they do, they'll be further transformed. We'll use those for in vitro fertilization as I described before. This has been done only in the mouse by our Japanese colleagues. And we know it's a huge leap from the mouse to the rhinoceros, but they have proved the principle. So we're confident that with sufficient time and technology development, we can be successful in the rhino. So the next question, of course, is, are these fibroblasts transformed to iPS cells and the tissue, are they functional? And this is a demonstration. Angelivu was the last male rhino that we had that died in 2014. These are his fibroblast cells, transformed to iPS cells and cardiomyocytes and beating in a natural rhythm in the Petri dish. This is Marisa Carodi, who has done all of this work in the genetics lab. So where does the southern white rhino come in? As I mentioned, because they are very closely related, we think they are excellent recipients and models for the northern white rhino. So the first thing that we did was we brought 6 southern white rhinos from South Africa from some private reserves, 3 different private reserves. We brought them in, we built a rhino rescue center at the San Diego Safari Park, and we brought them these 6 rhinos here. You can see their horns are blunted for ease in creating them and transferring them. So the day they hit the ground, we started collecting feces and doing progesterone analysis. The graph on the top is Victoria. We realized very quickly that she came to us already pregnant. So you can see the beautiful progesterone profile. Now it looks a little, it's not a very smooth profile. These are fecal progesterones. We don't bleed our animals on a frequent enough basis to smooth out this curve, but clearly she's pregnant. And then on the bottom left is Wallace. She's our smallest animal, and we assume she was the youngest. She also came to us with a severe bullet wound from a failed poaching attempt. But she cycles regularly and ovulates regularly. And what you see here, if you look closely at this profile, you can see that she has long cycles and short cycles. This is something that's unique to white rhinos. They do have short cycles that are about 30 days and long cycles that are about 70 days. And the long cycles were thought in the past, and it was even published, that they were infertile or subfertile. We know now that these are spontaneous, that they are normal. And actually we have established 3 pregnancies from artificial insemination following long cycles. It used to also be thought that they were failed early pregnancies, but Wallace had these long cycles, and she had not had access to a male the entire time we had her and prior to that. The other 4 animals that we brought in, if you see the profile down here on the bottom right, were not cycling. They were not ovulating. This is what we would see normally in southern white rhino females that are born in zoo settings, born in captivity. These F1s, first generations born in captivity. They often are flatliners. Less than 1/3 of southern white rhinos in captive situations reproduce. We have examined that problem and have solved it through dietary changes. So the first thing we needed to do with our animals was to train them. And so we have a team of professional trainers, wildlife care specialists, who train these animals to trust them. They come into a chute. This is not a squeeze chute. They are not restrained. Everything is voluntary. And I have to point out at the top, doing ultrasound with her happy face on. This is Parker Pennington, our postdoc. And thank you so much, Smithsonian, for sending us Parker. She's a wonderful postdoc. And we trade off on ultrasound duties. So what we saw was that these females were not ovulating. So the first thing that we did was develop an ovulation induction protocol. We were able to bring it down to a single IM injection of 4.2 milligrams of deslorelin, which is a GnRH agonist. And what we timed it so that we gave the deslorelin at about, when the follicle it grew to about 35 millimeters. And we could only do that because we had access to transrectal ultrasound on a regular basis. 3 to 5 times a week, we could ultrasound these animals and follow the follicle growth. So after we gave the injection, of course, we saw here, we saw a growth of the follicle due to the FSH and LH release, but then three different things happened. Either that follicle regressed and the cycle started again, or one of our females habitually forms a hemorrhagic anovulatory follicle, which is common in horses, and we see it in this female as well. In most cases, the animals ovulate. You can see this bright corpus luteum here that's easily visualized on ultrasound. We were able to, by timing the deslorelin injection to the size of the follicle, we were able to get 85% of our ovulations between 40 and 48 hours after injection. So then, to do artificial insemination, we start out with a standing sedation, which was not an easy thing to figure out for our veterinarians. Most of our, actually all of our immobilizations prior to this time have been full immobilizations. So to titrate the drugs to get to affect a standing sedation so the animal is calm, doesn't move very much in the chute. Again, she's not restrained, but also does not go down. It would be very difficult to do an artificial insemination on an animal that was sitting down. And we use frozen semen. So the catheter that we used was patterned after one I developed for the giant panda with a slight curve at the end. Of course, the giant panda catheter was much too short. We continued to elongate this catheter until now it is at 69 centimeters just to get through the cervix into the uterine body. The cervix of a rhino looks like this. It's very tortuous. It's extremely difficult to penetrate. And we are working with one of our local universities and the Advanced Robotics Lab, and they are designing a robotic embryo transfer and artificial insemination instrument that can actually navigate through this tortuous cervix. Here's the artificial insemination itself. So in the middle is Parker with her arm there holding the catheter in place. I am above doing a transrectal ultrasound to verify the placement of the catheter, but also to see exactly where the sperm is being deposited. And down below is Carly Young, one of our research associates, who's actually doing the insemination. So of the 3 pregnancies that we established with artificial insemination, the earliest one that we could actually visualize was 18 days. This one is 20 days post-AI. One of the 3 actually did not survive past 36 days. We saw that vesicle decrease and disappear. At 2 months, we could see a nice blood flow with Doppler ultrasound, and actually couldn't see any structures. Without the blood flow, it was hard to know what this lump of cells was, but it certainly was an animal or a fetus with a decent blood flow here. And then at 5 months, you can see over here on the left, swimming in the amniotic sac is a fully formed and growing fetus. And over here on the right at 11 months, so into the 2nd trimester, gestation in the white rhino was 16 to 17 months. So here you can see still swimming, same fetus. You can see the eye, the jaw, the mouth. At a certain period of time, at about the middle of the 2nd trimester, the fetus and the placenta are so large that they tip over the pelvic rim and we can no longer visualize them through transrectal ultrasound. So we have trained the animals to stand for transdermal ultrasound. And the results of these inseminations, our first one from frozen semen is Edward. He was 148 pounds at 494 days of gestation. And these gestation lengths are very interesting because field-bred rhinos, we rarely have an accurate breeding date because the male may follow the female for a couple of weeks. There may be breedings early in the morning or late at night when keepers are not there to observe. So they are at best guesstimates, but we now have exact gestation lengths. Our second offspring was born from chilled semen. Edward, the frozen semen, was only the 3rd southern white rhino ever born to frozen semen in the world. And actually both of these births were the first in North America for this species. Today happens to be Edward's 1st birthday. You can see in the bottom, Edward and Future greeting each other. The names seem a little odd, but they are named after donors. This is a donor and grant funded project. So next we are going into the lab and trying to figure out how to make these embryos when we do get sperm and eggs from iPS cells. Historically, we have only had post-mortem sperm and eggs to work with, but we are developing or have developed a very successful electroejaculation procedure for rhinos to get the sperm. We do full immobilizations, of course, for this. Our veterinarians intubate and insufflate, and we have 1 hour from the time the animal is darted, so we have to work fast. We set up a lab in the enclosure and do the initial analysis there, extend the sperm, take it back to the lab for further analysis, and either chilling for future artificial insemination or cryopreservation. Our first attempt to make embryos from post-mortem collected oocytes was back in 2004. And we did have semen that had been frozen for about 21 years. These oocytes were harvested about 10 hours after the female died. So very quickly in the world of post-mortem cell extraction, we were able to get 50% maturation and 30% with ICSI. We had 30% of the animals or the eggs actually developed 2 pronuclei. We did not get any cleavage from these, probably because the cytoplasm, this is a very old female, and the oocytes were collected so long post-mortem, so long after the female had died. So it's really a challenge to try to retrieve oocytes from living rhinos. There are 2 people in the world who do this. Luckily, we are associated with one of those people in South Africa. And so the depth of the ovaries prevents a transvaginal approach, which is done, of course, in humans, but also in cattle and routinely in horses. The thickness of the skin and the position of the ribs also prevents a transdermal approach, which was used originally in cattle. But the proximity of the ovaries to the rectum and how we can visualize them with ultrasound does allow for a transrectal approach. So this is, we were visiting South Africa this year, and we did OPU on several rhinos. We also brought this technique back to San Diego and did OPU on 4 of our rhinos. This is the instrument up here. This is the OPU probe. And it's quite large, as you can imagine. Here it is in operation. It consists of an ultrasound. It's ultrasound guided. There are 3 tracks with 3 needles. So because we cannot manipulate the ovary, so we can't turn it around to get to follicles on all sides. So we have 3 needles that we can puncture 3 oocytes or follicles at 3 different levels. So this is what they look like. This is a typical OPU collection from a rhino. The number of oocytes collected ranges from 0 to 13, average probably 6 to 8. And then our challenges are in vitro maturation and embryo development. One of the things that we are working on now is somatic cell nuclear transfer as an option while we wait for those sperm and eggs to be developed through the fibroblast cells and iPS cells. So we started, we were very lucky in that we had Dr. Marta Gomez in our lab for a while, and she is the domestic cat cloning queen, and she was in our lab and helped us very quickly learn how to clone domestic cats as a model. We then moved to cloning domestic horses, which was a little bit more challenging, but we can routinely clone horses now. We have used horse oocytes, domestic horse oocytes. Let me back up a moment. The horse is the closest domestic relative to the rhino. And reproductively, many of their features are very similar. Not all of them, but many of them. So the horse is the best model that we have for the white rhino. So we have used enucleated domestic horse oocytes and transferred or fused either Northern White Rhino fibroblast, Sertoli cells, or iPS cells. and we were able to fairly routinely create embryos. However, they are mitochondrial DNA chimeras. So this is not acceptable, obviously. And so a big focus for us right now is to eliminate, deactivate, or flood the equine mitochondrial DNA and to see if we can use interspecies embryo transfer. We've put together a wonderful team, many of whom might be on the call today, to help us work with the mitochondrial DNA. This is a new area of research for us. So even though our focal animal is the Northern White Rhino, these techniques, if we can perfect these techniques, they will have great application to all the other rhino species that are all endangered. The black, the Sumatran, the Javan rhino, critically endangered, very low populations of Sumatran and Javan rhinos. Assisted reproduction will almost certainly be necessary to save these species. But in addition to rhino species, this highly collaborative, interdisciplinary approach to reproduction, solving reproductive problems could be applied to any species of any taxon. And we hope it will be a model for conservation research going forward and reproductive research going forward for the protection of endangered species. So I wanted to thank our collaborators. These are our collaborators in South Africa. And we have formed a group called the International Rhino Reproduction Collaborative. We meet monthly and we meet in person in South Africa every 2 years. They have been wonderful collaborators and they have so much expertise with this species, Southern White Rhino, and also have rhinos that we can work with hands-on in the field. And also, of course, my own group and our wildlife care specialist without whom we would not have the access to the Southern White Rhinos that we do to learn in great detail about the reproductive processes. Our veterinary staff who provides the support for all of our procedures, our own reproductive sciences group, and so many collaborators. Here are just a few. Thank you very much.